here we are at the IsoLab. Go ahead and see if Andy is ready for us. Hey, Andy.
Hello, welcome to IsoLab. I'm
All
Andy
right.
Schauer, the lab manager here. And we're going to do a tour today.
Awesome, thank you. All right, wow. Let's go on in.
The IsoLab supports researchers from a number of different departments and programs at the University of Washington, as well as off-campus collaborators. During this tour, we will learn about the wide range of isotopic analyses and sample preparation methods that are available in the IsoLab.
Basic things we have in the lab: refrigeration, drying ovens, a muffle furnace, chemical fume hoods, of course. Okay, we are in the sample preparation side of IsoLab. Over here, any samples that we bring in from the field, oftentimes we'll need some sort of preparation. That could be as easy as moving water from one container to another, or weighing sample material from a field container into a sample capsule. Or it could be something more complicated, like in this case, we are injecting water into a cobalt fluoride, hot cobalt fluoride column, which fluorinates the water, converts it into HF and O2, and then we freeze the O2 and measure the oxygen isotope composition of that, getting the O16, O17, and O18. This is useful for lots of ice core projects that we have in the lab, where you're trying to not only estimate the temperature when the snow fell before it was an ice core, but also the O17 allows you to say something about sea ice extent or the conditions when the water was first evaporating over the ocean. So some more subtle detail questions can be answered with the O17. That we've just created, we then transfer it to a cold finger. So now this is pure oxygen that we can then carry from this preparation line over to the mass spectrometer to measure the oxygen isotope composition. We'll see that mass spec in a little bit. An example project that uses O17 of water from this system comes from Eric Steig's research. And he's a polar scientist, and he uses O17 from ice cores to talk about various paleoclimate applications. Another more complicated preparation system we have on this side of the lab is a relatively new prep line that takes CO2 that has come from carbonate and oxygen from a cylinder. It equilibrates it in a furnace that is held at 800 degrees Celsius with platinum. That makes the CO2 oxygen and the oxygen atoms exchange with each other, and then we purify the oxygen out and the CO2 out to measure on a mass spec that we'll see in a little bit. And that allows us to get the oxygen 17 of carbonate, which is an up-and-coming method that might help say something about monsoon seasonality or relative humidity or evaporation in paleoclimates. Another sample preparation line for rock samples that have very low nitrogen concentrations. We can't use a traditional elemental analysis like you may have seen, and we're also going to see later today, but this particular case we do an in-tube combustion, so we put all the reagents and the sample material in a tube, pump away all of the atmospheric gases. That goes in the muffle oven at a very hot temperature, and then it comes back over here and we purify the nitrogen gas that has now been created away from all of the combustion ash and everything, and then that tube goes over to a mass spectrometer in the other side to measure the nitrogen isotopes of very old rock samples.
Okay. And can you give me an example of where one of those rocks in the world might have come from, or what question you would ask?
Right, so questions about the pH of very old lakes when biology was first getting going and was it a high pH scenario, and that would have left an imprint on the nitrogen isotope composition. Or, so these rock samples are coming from very old rock outcroppings in Antarctica or in Australia or other places around the planet. A lot of the samples or the preparations we've been talking about so far, they take a long time to make even one sample. And this example is probably the slowest sample preparation and analysis that we have in the lab. You can do about one sample per week. There's a lot of back and forth to the muffle furnace and back and clean up in here before you can even take it into the mass spectrometer.
Billions of years old rocks take a long time to,
yes,
to prepare.
Yes. One of the researchers that uses this very low nitrogen old rock technique and preparation is Roger Buick, who's interested in when biology was first getting going on Earth. We also have a bacteria denitrification method set up in the lab. This is designed to measure the nitrogen and oxygen isotopes of nitrate. And so there's a lot of bacteria prep that happens in here. This is the species that we use, Pseudomonas aurifaciens, and they are handy for this technique because they convert nitrate to nitrous oxide as a denitrifier, so they stop at a place where we can measure the nitrogen and the oxygen isotopes. Many denitrifiers will take their nitrate all the way to N2, and that isn't useful for us if we're trying to measure the oxygen isotopes. So there's a lot of media work in here and other bacteria work that we have to accomplish on this side before we can take samples to the mass spectrometer side. The types of samples we would analyze with this method are if you're trying to get a handle on the nitrogen cycling for a certain ecosystem, then you might want to know what the water in that ecosystem, what the nitrogen backgrounds are for that, or maybe you are interested in knowing where some higher levels of nitrogen in your water system are coming from. Is it a pollutant? Is it from fertilizer on somebody's yard? And so that can help you pinpoint where certain nitrate sources might be coming from. This is our carbonate prep line for clumped isotopes, and in this prep line we start with a carbonate sample, and that carbonate sample falls into a hot phosphoric common acid bath. That's like the volcano reaction that you may have done when you were in elementary school, except in this case we actually want to save what is coming out of that. So water and CO2 are coming out. We freeze the water, the CO2 keeps going, and we freeze the CO2. Because this is clumped isotope carbonate work, we need to be extra clean, so there's a step where that carbonate is cleaned, the CO2 is cleaned, and then we freeze any water out again that may have made it through, freeze the CO2, and lastly we end up with moving the CO2 from the carbonate into a Pyrex brake seal tube. We flame seal this, and this is the purified carbon dioxide that ends up going to a mass spectrometer on the other side.
And how long does this whole sequence that you just described, how long does that take per sample?
This takes about an hour and a half per sample over here and about 3 hours per sample on the other side. One of the applications for our clumped isotope system in the lab comes from Kate Huntington's research group. Okay, now that we have our samples prepared, we are ready to take them into the mass spectrometer side, so we're going to go over there. Okay, so now we're in the instrumentation side. Maybe you can hear the white noise through our microphone. We have 5 mass spectrometers. All of them are pretty specific in what we have them set up to analyze. We also have 3 laser spectrometers that are all set up to measure water isotopes. This is a traditional light stable isotope lab, which means we can measure hydrogen isotopes, and it's stable isotopes, so no radioactive isotopes in here. So hydrogen, nitrogen, oxygen, carbon, and sulfur are all of the isotopes that we can measure in this lab. There's even some on the other side. Okay, we're going to start with our newest mass spectrometer that measures carbon dioxide isotope composition. And this one is special because it measures all of the masses of CO2 for our clumped isotope application. So a lot of our mass spectrometers have this thing poking out the top. This is the flight tube. The carbon dioxide comes in here. We turn it into an ion in the source. This is the electron bombardment source, where the gas that we're interested in enters through this little hole. In the case of the instrument we're standing in front of, carbon dioxide or nitrogen diatomic nitrogen. That gas enters into an ionization chamber where one electron is knocked off. Now it has a positive charge, and it's pulled out of that chamber and focused and accelerated down the flight tube. So this source is sitting right here inside the flight tube. The ion gets propelled down the flight tube. Once it starts to feel the force of the magnet pushing it towards you, it pushes the lighter isotopes towards the center, and the heavier isotopes have a wider radius, so they go a little wider, and in doing so we end up with all of the masses of CO2 in this case separated from each other, and then they're measured in a Faraday collector at the end of the flight tube. Once the preparation is done with the bacteria in the preparation side of the lab, we end up with a vial like this, which has the media and the bacteria in it. It did have nitrate in it. All the bacteria converted that nitrate into nitrous oxide, which is now in the headspace. That goes on here, and an auto sampler will purge these vials with helium. And in that helium stream are all kinds of other gases that we're going to try to get rid of. So we're getting rid of water and carbon dioxide. And then that nitrous oxide freezes out of the helium stream with a liquid nitrogen dewar here. When that part is done, we have a frozen bit of nitrous oxide in the helium stream. We let that warm up, and that nitrous oxide goes to the mass spectrometer to get the nitrogen and the oxygen isotope composition. Here we are at our third mass spectrometer. This one looks like the first one that we looked at, where you can see the flight tube poking out of the top. And this one is designed to measure oxygen isotopes of O2. And so in the other side we saw preparation lines that either fluorinated water to make O2. Those come in, those O2 samples come in to cold fingers. Here's a manifold of 8 cold fingers. We could also create O2 from the other side on the prep line where we saw O17 of carbonate. So all of the oxygen that is purified in the other lab is brought to this side and measured for oxygen 16, 17, and 18. It also has another peripheral attached to it, 2 more peripherals. And these are designed to do more of the purification for us. So One of our researchers, Becky Alexander in atmospheric sciences, is interested in atmospheric chemistry, and she uses nitrate and sulfate from aerosols to talk about different aspects of ozone abundance in the atmosphere and the oxidative capacity of the atmosphere. And she uses this instrument to measure the oxygen isotope composition of nitrates or sulfates. And so this TCEA is a space where that reaction, a silver sulfate or a silver nitrate capsule will fall in here and pyrolyze. So that's just heat, no real reaction, just heat breaking apart, decomposing a molecule, and it turns into NO2 or NO and SO2, and then the O2 continues on getting purified and in a helium stream into the mass spectrometer.
And what temperature does that happen at?
It depends on the but if it's sulfate, this happens at about 1,000 degrees Celsius, and if it's nitrate, we can do it a little colder at 500 degrees Celsius. SHREC, our mass spectrometer that we use to measure carbon and nitrogen and sulfur isotopes. So with all of those different elements, we have different ways to introduce samples. The most complicated, as I mentioned in the other side, where we were preparing very low nitrogen concentration rock samples. When those samples are ready for the mass spec side of the lab, the tubes are put into this tube cracker, which then has a helium stream running through it, and they're cracked. Those glass tubes are cracked, and the nitrogen is frozen into a loop, and then that loop is thawed and the nitrogen is released into the helium stream and for the nitrogen isotope composition measurement, nitrogen 14 and nitrogen 15. Our carbon and nitrogen elemental analyzer, and so in this case, samples are put in the carousel, they fall and combust in a combustion column. Carbon dioxide and nitrogen are purified from other gases, and they're separated from one another. The nitrogen goes into the mass spectrometer for nitrogen isotope analysis, and then the carbon dioxide goes in, and in this case we're only interested in the carbon 13 and carbon 12. This is our sulfur isotope composition elemental analyzer, EA, and this one is specifically for samples that fall into the EA. Those combust, we create SO2 and purify that SO2 away from anything else that's present. And we've just added a recent addition to this where we freeze the SO2 in a liquid nitrogen loop, and then that tightens up the SO2 peak. So we can measure smaller samples from ice cores, for example. Yeah. This is the combustion column that is used for carbon and nitrogen isotope analysis, and that's at 1,000 degrees Celsius right here. So the sample falls and the flash combustion takes place right here. The gases that come out then go through a reduced copper column. So they come out down here and go up through the reduced copper column. This ensures that all of the oxygen that we were using for combustion is absorbed, and it makes sure that nitrous oxide is converted to N2. This is our fifth and final mass spectrometer in the lab. This is porous, and we have this set up to measure carbon and oxygen isotopes of carbonate, which is different from the very first mass spectrometer that we saw. This one only measures 13C and 12C and 18O and 16O, and it does not do the clumped isotope measurement. It has its own carbonate purification system attached to it, so we can load carbonates into vials and it will drop phosphoric acid onto each carbonate in the same way that we saw on the prep side. So we have a sample purification device attached to Boris. This is the purpose of this is to turn carbonates into carbon dioxide, just like we saw on the other side. Except in this case, we put carbonate into little vials, and we load up about 50 of these vials that can run overnight. Phosphoric acid is dropped onto each carbonate sample. That makes water and CO2. The water and CO2 are separated from one another, and the CO2, the carbon dioxide, goes into the mass spectrometer for carbon and oxygen isotope composition.
And why does it have its own box?
It has its own box because it's a lot of extreme temperatures. We have a hot reaction temperature. We need a space to put the vacuum pumps to pump away all the atmospheric air, as well as liquid nitrogen, and where all of the freezing of the gases happens. Do you have one of those yet? This is a water isotope laser. So this measures the hydrogen isotope composition and the O18 and O16 of the water. Water is pulled up into a syringe and injected into a vaporizer, and then that water vapor goes into the laser for isotope composition measurement. If you've had general chemistry and you worked with a spectrophotometer, where you put in the little tiny cuvette and it shines light through the cuvette so that you can measure the concentration of whatever solution you have, this works in almost the exact same way, except our cuvette is about this wide, and the path length, because there's mirrors on either side, and our energy source bounces back and forth so many times that our path length is about 1 kilometer. And that gives us nice resolution to measure the isotope abundance of, in this case, water. So each one of these peaks is an injection from the syringe, and this is showing the water concentration for each peak. And then this is showing the oxygen 18 composition and the hydrogen isotope composition. So you can see the change from a more Antarctic type water here at -200 to something that is more local to Seattle at maybe 0 to -10. This is Phoenix, another water isotope laser. This one is very similar to the one we've already seen, except it also measures the O17 in the water. And that means with O17 being in much lower abundance on planet Earth, we have to measure the O17 in water injection for a lot longer. So these injections happen very infrequently, and we let those peaks sit in the instrument for a lot longer to get a more careful measurement. Just like the other water isotope laser that we saw, this is a cavity ring down instrument where the infrared energy is shined through a cavity and bounces back and forth, making our effective path length about 1 kilometer.
Okay.
We're gonna prepare a couple of samples for carbon and nitrogen EA analysis. We use tin capsules. Zero the balance. This is our salmon reference material. It's Bristol Bay sockeye salmon. It has a very high, for animal tissue, nitrogen isotope composition, and it's very homogeneous. We end up using it as a reference material. put in there.
Filming again.
Okay. Ball up your sample so that it fits in the auto sampler. Okay, now we're going to load in a couple of EA samples. I'm opening up the auto sampler. Want to come and see before I close it. Connected to a vacuum pump, so we're pulling all of the atmospheric air out of here right now. Okay, here's one analysis from the salmon sample, and what you're looking at is time on the x-axis and millivolt readings on the millivolt signal on the y-axis. So this signal comes from the Faraday cups that we talked about before. We start off with nitrogen, measuring N2, and we first see a reference peak. This comes from a compressed gas nitrogen cylinder. And then the salmon nitrogen peak comes out. It looks a little more Gaussian. And once that peak has been detected, there's the instrument switches to carbon dioxide. And then the salmon carbon dioxide peak, followed by a carbon dioxide reference peak from a compressed gas cylinder. The whole trace takes a little over 700 seconds, so here's 600 or 10 minutes right here.
Hi, my name's Anna Sullivan. I'm a first-year PhD student here at UW. I'm working with advisor Kate Huntington. So I have been working on a project with the USGS. We are using carbonate samples to understand the paleoclimate in Las Vegas Valley, so in Nevada. There we use the carbonate samples to see how cycles of wet and dry climate affect the valley and the fauna and flora that live there. So this is a schematic of our prep line. All of the valves are controlled by this computer. So the sample starts here and then ends up at the brake seal over here. So the sample starts here at the very top, and then it drops through this tube into a potoform acid. The carbonate then dissolves into CO2 gas, and it starts to move through the prep line. So the prep line is there's different temperatures in all of these different dewars, and things like water and impurities get trapped in those, so that the CO2 gas can move through by itself, and then it'll be ready. Okay. You probably started. And then end up in this glass tube. And then once the CO2 ends in this glass tube, I flame seal it closed, and it ends up just like this. Okay, so once we get our CO2 gas in the glass tube, we label them and then we bring them over to Polly.
Hi, my name is Lindsay Davidge. I am a fourth-year graduate student here at the University of Washington.
I actually obtained dual bachelor's degrees from the University of Rochester in chemical engineering and in environmental science. And some really exciting undergraduate research actually led me in the path of pursuing research as a career option. I actually had the opportunity to go to the center of Greenland the year after I graduated. And that sort of set a spark in me for polar climate experience. I actually worked as an engineer in Seattle for a number of years before realizing that it was really that research path that I wanted to be pursuing. So that led me back to graduate school, where I'm now enrolled, and have this exciting opportunity to continue working with ice core samples. The samples that I'm working with are actually from the very site that I traveled to as an undergraduate. So this is very exciting for me. So I am broadly working with various types of data, both geochemical and geophysical, that we use to better contextualize and understand the measurements that we can make from the ice cores themselves. So I'm interested in understanding the processes that change the ice itself, so that when we get those long-term climate records, we're able to disentangle the signal with better fidelity. Okay, so before I mentioned that I'm using a diverse array of tools to contextualize ice core measurements, but actually the first project of my PhD has involved making direct measurements from ice cores. So what I have done in this freezer is create a system that will continuously measure an ice core so that we can look at each individual layer. And that allows us to construct really high resolution records of past climate data. So let's head into the freezer and I'll show you exactly how that works. Great. This ice core here is from Summit, Greenland, so in the center of the continent where I pointed before. And it's really cool to be able to work with these samples, because these are formed, so these are collected vertically from the ice sheet. I don't know if you've talked about this. And as snow falls continuously on polar ice sheets, snow compacts and densifies such that each horizontal layer like this is an individual layer of annual snowfall. So we are able to take these vertical samples from the ice sheet and then measure them in that same orientation, so that we can measure from bottom to top and get a record from old to new of what the geochemical signals look like in that ice sheet. That we have made, we cut them in a different shape when we actually measure them, but this is useful for seeing some of these vertical features. I don't know, I can hold it up to the light. We might be able to see a few more layers. There are some. So when we're measuring an ice core, we actually cut them into a thinner shape that is more similar to the square of this tube, and we will actually set it inside the tube. And I then mount them on this tower here. Sorry. Where we are able to align them with a melt head. So right here at the top of this box, there's a heated element that was actually designed by the Center for Ice and Climate in Copenhagen. And it is designed to melt the bottom of the ice core, and it takes the melt from the very center, which is clean and untouched, and it sends it through the wall to our instrument. And the melt from the outside is also sent through the wall, but it's not analyzed because it's been handled and might have impurities in that ice. So this tube is used to align the core so that while it's melting, we're getting each horizontal layer and not something crooked that might be mixing annual signals. So inside this box is where some of my graduate work has been conducted. So this is just an insulated box, and as you can see, there are different tubes that collect each one of those streams. There are also heater cables here. So we have heat cartridges that are sitting inside so that we can keep the melt head at a very consistent temperature, so that we can control the rate of melt and make sure that we're not causing any changes to the ice, and we're not causing any fractionation while we're controlling this process. So each of those tubes goes through this insulated pipe, through the wall and to the other side, and we can actually open this door, and we can see the setup that's on the other side. So during analysis, we're able to actually look through this wall so that we can communicate with folks on the other side and make sure that everything is happening smoothly. the pipe that we saw on the other side comes through the wall right here. You can see there are various heat cables and the sample tubing itself that's drawing the sample out through these two pumps. So on the right hand side we have two peristaltic pumps and they're pulling ice from the melt head and then pushing it through a series of filters that are actually removing particles so that we can better control the flow. If there's a layer of really dirty ice, especially if there was something like a volcanic event or some other reason why we might have a lot of fine particulate, we want to make sure we remove that or it's going to clog our system and make us incapable of making good measurements of the layers that follow. When this is running, the most critical step of our sort of pre-analysis sample processing is actually turning the sample from a liquid melt into a vapor. So the laser spectrometer instruments that we use, I know you talked earlier and saw the L2140, which measures triple oxygen isotopes of water. So we're looking at delta 17O, delta 18O, and delta deuterium of the water that's the actual ice matrix of the ice core samples. But we can't measure them as ice and we can't measure them as liquid. We have to turn it into a vapor so that it can flow through that instrument. And so in order to vaporize, we want to do that in a way that doesn't fractionate our sample, or we won't be able to make a true measurement of what it was in its original state. So once the particulate has been removed from the sample, we actually also remove bubbles from the sample. Glacial ice is about 10% air, and so we want to remove all that air so that we're not just so we're not measuring that air mixed with the water sample, but we're actually just measuring the water itself. And after the air is removed, the sample continues through to this final filter, which is typically plugged in to this box. So you can't see what's happening in here right now, but you can see that it has two electrical connections. So it is actually heat controlled by a PID controller here. So this is keeping it at a very constant temperature. And then this wire is actually heating it so that we can hold it at 170 degrees Celsius at all times. And that gives us a really constant condition for our vaporization. Great. So as I mentioned before, from our continuous flow ice core analyzing system, we are measuring delta 17O, delta 18O, and delta deuterium at the same time. That actually allows us to determine these additional quantities, such as deuterium excess, which is the difference between the delta deuterium and sort of what we expect delta deuterium to be based on the relationship to O18, and the oxygen 17 excess, which is similarly the difference between the measured oxygen 17 and the expected oxygen 17 or 17 based on the typical relationship between O17 and O18. And so if we're able to measure these additional records, these actually give us additional data points that we might be able to use to better understand past climate. So you've probably heard that delta deuterium and delta 18O are commonly used as temperature proxies so that we can make these measurements and look at a past record of temperature based on what the delta 18O or delta deuterium was doing in the snowfall. But if we're able to make these additional measurements, such as oxygen 17 excess, we actually have an additional data point that might be useful for improving climate models and really better understanding what those temperature records are telling us about the past.